Okay, so we are going to be talking today about regulation of cell division. This lets me do this. All right. So as we're going through this process, we talked about mitosis yesterday and all of that good stuff. But now, today, we're going to look at basically how this actually happens and what happens if it goes bad. So when we're coordinating all these different things for the cell cycle, we need to be able to do it properly every single time. One of the reasons why is because if we do it improperly, we're going to get baby cells that don't have enough DNA, they have too much DNA, or they're dividing out of control. Not all cells, really, really important on that bottom line there, have the same cell cycle. Make this bigger. Ooh, wow. It's gonna make noises. I don't want that. Okay. So when we're looking at the frequency for cell division, if we're looking at like an embryo, so when the mommy and the daddy love each other very much, when we fertilize that zygote, every 20 minutes, we're gonna make a brand new cell. It happens very, very quickly in that process. Whereas your skin cells divide once every 12 to 24 hours. And the reason why they have to happen so quickly is because your skin is constantly flaking off. We have constant abrasion happening. Liver cells are really, really unique in our body that they contain the ability to divide throughout your life, but they only divide about once every year or two. This is really, really important for a couple different reasons. One of them is for people to get liver transplants. So you can actually cut part of your liver out, put it in somebody else, and they can grow a brand new liver and you can heal your liver. It takes a while to fully get that part back, but it can regrow. This is also really, really important because your liver is a detox organ in your body. So if you go through a period of your life where you're putting in some not good stuff, or if you're going through something like chemotherapy, where you're taking a bunch of drugs and medications in order to get rid of cancer, your liver can actually regenerate itself to heal those tissues that had to work so hard during that chemotherapy process of like detoxing your body. Mature nerve cells and muscle cells don't really divide at all after maturity. However, that was kind of an old school of thought. Now we're getting some research that they can, in fact, divide after they mature. It's just a very, very long cycle. So some nerve cells can take up to 25 years to actually regenerate. And we're actually seeing a lot of work done with stem cell therapy, like with severed spinal cords or different things like that, where we can put in actually stem cells to help those nerve cells regenerate to regrow parts that were disconnected. So when we're talking about the cell cycle and the controls, there is something called a checkpoint. There are two checkpoints in the cell cycle as a whole that are irreversible. So when we replicate the genetic material, we can't go back and unreplicate. And when we separate the sister chromatids, we can't unseparate them. So when we're looking at these, your cells are going to make sure that those chromatids got separated, and it's going to make sure that that genetic material got copied correctly. With these checkpoints, they are the stop and go point, and they're going to send out chemical signals to the rest of your cells to indicate if it happened correctly.
Did you say it was the separation of chromatids or chromosome? Oh, okay.
The chromatids from each other. Yeah, so that's another one of those checkpoints. Really good question. Yep. And when we're looking at these, there are three main irreversible checkpoints. The first one is going to be in G1, between G1 and S, and that is, can DNA synthesis begin? Did my cell grow large enough to warrant actually going through cell division? So like if my fist is the size of a normal cell, we want it to get maybe at least double that size before we divide. Because if we divide a cell that's not yet big enough, we're going to get too little, like the people that have little baby hands, like those creepy little things, those creepy little plastic things. We don't want the cells to get smaller and smaller and smaller. We want them all to be the same size. So that is our first checkpoint, to make sure enough growth has happened. Our second checkpoint is going to be between the G2 and the M phase, which is actually the process of mitosis. So we're checking, has DNA synthesis happened? And now once we start, we are committed to mitosis. You can't undo mitosis. Ladies, it's like putting on a hat. Once you put on the hat, you got to commit to the hat, because otherwise your hair is going to be all a mess, and we don't want that rat's nest to be out in the world, right? So we have to commit to mitosis. Once mitosis begins, we do have one more checkpoint, and that is the spindle checkpoint. So if we remember from yesterday's lecture or the lecture about mitosis, when those spindle fibers attach to the centromeres at the kinetochores, we are making sure that all of those spindles are attached. Otherwise, what can actually happen is if we have like our sister chromatids and the kinetochore doesn't form on one side, both of those can get pulled over to one cell. So one cell is going to get an extra chromosome, and then the other cell gets one less chromosome. When this happens very, very early on, like in fertilization, this is actually one of the ways that Down syndrome can happen. So mistakes do happen, but typically what our cells will do if they find a mistake is they kill the cell in a process called apoptosis. So when we're looking at these individual checkpoints, we're going to kind of go through each one. The G1S checkpoint, that is our primary restriction point. If the cell gets the go signal, then we're going to start growing, and it's going to send out growth factors to other cells around it. If the cell doesn't receive signal to grow, it's going to turn into the G0 phase, or just like the hanging out phase. Your cell's just vibing, hanging out, if it doesn't receive those external signals. When we talked about cell signaling, one of those signals that a cell can receive on the outside is a growth factor that can be sent from another cell that says, hey, you guys all better start growing and start dividing because we need to heal this tissue. So like if you get a big cut on your arm, that can actually send out growth signals, like your white blood cells can send out growth signals to all of the other things around that wound that say, hey, you guys better start growing so we can repair this wound. When we're talking about the G0 phase, this is like ultimate interphase. It's a non-dividing, differentiated state cell. Most of your cells are in the G0 phase. They're just hanging out. Nerve and muscle cells, they are called arrested in the G0 phase, and they can never divide again. Once again, we are finding new research that is not always the case, which is very interesting. And the really cool thing about science, that we're always discovering kind of new stuff. So when we're activating the process of cell division, we have cell communication signals that get sent, and the signals are those ligands that are proteins that get sent from one cell to the other. These signals can either be an activator or an inhibitor. So when a cell sends out a signal, it can activate other cells around it to start growing and dividing, or it can inhibit cells around it to start activating and dividing. And this is also one reason why some chemotherapies work, because they actually send out inhibiting factors to tumor cells to say stop growing. If they get a go-ahead signal, what will happen internally is they will send out promoting factors that will then tell that cell to start dividing. And it goes through a whole phosphorylation cascade. So that's why we talked about cell signaling first. So these kinase enzymes will either activate or inactivate cell signals. With these cell signals, they are something called a cyclin kinase. Your activity that you guys are going to do next Monday goes more in depth with this, because this is actually some different ways that cancers can be caused. So we're just going to like mention the terms here, and then in your activity we're going to do on Monday, it goes a little more in depth with this. So a cyclin is a type of regulatory protein, and this levels cycle in the cell. CDKs are cyclin-dependent kinases. So remember, kinetic, that's like a movement thing. So when a cyclin attaches to a cyclin-dependent kinase, here's where we can actually turn on or turn off cell division. That whole complex is then going to trigger passage through the cell cycle. So it's like this whole like mix of enzymes and cell signaling and all of this different stuff that actually turns on and off the cell cycle. When we're looking at this graph up there, so we have like relative concentration of cells over time, the cyclin activity is going to spike during mitosis, and then after that it goes way down. When those cyclins start to build up again to a certain point, it's going to activate different stages of the cell cycle. And these are just the scientists that kind of discovered that this happens. So just one way that cells regulate themselves. So looking at all of these checkpoints again, so we have the CDK cyclin kinases that are activating in G1 and G2. And then if we're looking at the spindle phase, we have an APC, and that is going to be a different complex. So it's the anaphase promoting complex that is going to either let anaphase happen, where we are splitting our chromosomes apart, or it's not going to let anaphase happen. So this is going to happen in that metaphase part of the cell, and it's either going to let it go on, or it's going to stop that whole cell cycle and kind of kill the cell. If it's really, like if those spindle fibers are all over the place, it's not going to activate that APC, and it's not going to go on to the next phase. So once again, these just drive the cell cycle from one to the next. And the genes for these proteins have been highly conserved. So the genes for all of these kinases are really, really similar in bacteria and plants and yeast. And this is one of the reasons why they always say like, oh, we can't cut down the rainforest because we might find the cure for cancer. All of these genes for those cyclins are very, very similar, whether you are a person or whether you are like an oak tree. They're very, very similar, which is very cool. So one of the ways that cancer is caused is when your cells ignore those cyclins and they're like, Mom, you can't tell me what to do. I'm going to divide whether I want to or not. And that's how tumors are formed. So if we can actually find maybe some different plant therapeutics that can restore this activity or something like that, it could potentially be the cure for cancer, which is kind of cool. Some other things externally, so that's like an internal thing that controls cell cycles. So just know that that's like an enzyme complex that controls whether the cell goes from one cycle to another. Externally, cells kind of like go into the space that they're in. They don't like overflow their space. So think of your body as like your cell's space. They're not going to overflow your body because that's what a tumor is. So these are external growth factors, and they are touch factors. So we have density dependent and anchorage dependent. So density dependent inhibition is if you have cells, so if you look on the top, it's like a petri dish with a bunch of individual cells on it. They are going to grow until they all just touch each other. Once they touch each other, all of those external glycoproteins, glycolipids, all of the things on those membranes, when they come into contact, that is going to trigger the cell to stop dividing. So that is going to act as a ligand that's going to shoot a phosphorylation cascade that's going to tell the cell, hey, stop dividing. I'm touching somebody else. I can't divide anymore. Anchorage dependence is because cells have to be anchored to a substrate. So if I have like a, oh my goodness, like a flask full of broth with a bunch of cells in it, the cells that are going to divide are going to connect to the glass. They're not just going to divide in the broth inside of that substance. They have to be attached to a substrate because they have to be touching something else in order to divide. This is one of the reasons why tumors will grow on other tissues and not just like in the middle of all of the acid in your stomach or like in the middle of your blood or something like that. So cancers have to be touching something else. But if they're not, that's what forms cancer, which is kind of weird. So when we're looking at these growth factors, we have our ligand hitting our protein, and we can see our phosphorylation cascade. So what that's going to do is it's going to send a message in the nucleus of your cell to your chromosome that's going to turn on a gene for that CDK, for that cyclin kinase, that says, hey, let's do cell division. Or with what you guys know about cell signaling, we can turn off that pathway and say, hey, you know what? Don't make any more cyclin kinase right now. We're touching something else. The cell doesn't want to divide anymore. So an example of one of these growth factors is PDGF, which is platelet-derived growth factor. So platelets are in blood, and they're made out of the platelets in blood clots. When we bind this PDGF, it simulates cell division in connective tissues. So what happens is if you have like a torn ligament, one thing that we can do is actually like inject growth factors into that ligament to help your ligament to heal. Or down there at the bottom, we got good old Lance Armstrong, who took some naughty things and he got all of his Tour de France titles taken away from him. So there's something called EPO, or erythropoietin, or biotin. I can never say that word. But what EPO is it's a way for athletes to blood dope. So typically your red blood cells will replace themselves about every 2 weeks. However, if you take EPO, that increases the rate of the cell cycle in your blood cells, and they will replicate more quickly. The more red blood cells you have, the more oxygen you can carry in your blood. With what you guys know about cell respiration, the more oxygen you have, the more ATP your mitochondria can make, and the stronger, faster, the more energy you actually physically have in your body. There have been many, many athletes, especially like extreme endurance athletes, like marathon runners or stage cyclists, like Lance Armstrong was, dope with EPO. And it is very, very illegal to do this because it's giving you an unfair advantage because your body can actually make more red blood cells. And this is just one of those growth factors. However, some of these growth factors can also create certain types of cancers. So some growth factors that we have are like human growth hormone. Those will also stimulate your cells to grow. However, if we have a proto-oncogene, proto like a prototype, that means first. So the first oncologists are cancer doctors. So onco means cancer genes. So the first cancer genes, these will activate cell division. When they're switched on, they can cause cancers because they ignore cell cycles. They say, I'm going to ignore this. I'm going to replicate however I want. I don't care if my DNA is damaged. I don't care if the spindle's attached appropriately. I'm just going to keep replicating, whether I am touching another cell or not. Your body has natural tumor suppressor genes. If these are turned off, they can cause cancer. So the big famous one is the gene P53. If your body has the gene P53 that is faulty or turned off, that will create many, many types of cancers. Very specifically, it is one of the main reasons why women and men too can get breast cancer, is because they have a faulty P53 gene. When we're looking at cancer and cell growth specifically, cancer is a failure to divide controllably. So cancer is uncontrolled cell growth. We're losing checkpoints. We're not turning off cell division. We aren't shutting down tumor, or we're shutting down tumor suppression. All cancers have to be able to shut down P53 activity. Every single cancer. That is the cell cycle enforcer. When we're looking at a normal versus an abnormal P53 gene, that normal gene in the first step, if we have some sort of DNA damage, cell division will actually stop, and P53 will trigger enzymes to repair the damaged section of the DNA. P53, or it will destroy cells that are damaged beyond repair. In an abnormal gene, if we have some sort of damage to the DNA, P53 fails to stop that like repair to the damage, and then we're just going to keep making damaged cells, which eventually can become cancerous. This whole big list of things are 6 different ways and 6 key mutations that happen in order for cancer to occur. So if we turn on a growth promoter, that's going to cause unlimited growth and create a tumor. So a growth promoter, promoting means like, yeah, you go. That's going to cause unlimited cancer growth. If we ignore the checkpoints, so if we turn off tumor suppressor genes like P53, we're going to ignore those cell checkpoints and we're going to make copies of damaged DNA. If we turn off the suicide genes, we're going to be able to escape the process of apoptosis. So apoptosis is programmed cell death. This is a very, very good thing for our bodies because normally if we have damaged cells, your body's just going to kill that cell. If we turn on chromosome maintenance genes, cells are going to live forever. We think of that as being a good thing, but eventually we want the cells to not live anymore. It's basically just like Little Penguin says, it's like out of control car with a bunch of systems failing. So it's like trying to drive your car as fast as you can and your muffler's falling off and a hubcap falls off and your rear view mirror falls off and you just keep driving as fast as you can. If we promote blood vessel growth, that is going to feed that tumor, give those cells more energy to divide even faster. And if we overcome anchor and density dependence, so if we turn off those touch sensors, then we're going to create cancer cells. So if you remember the slide we looked at before with like the little petri dishes with cells, the one up in the corner here has cancer cells that are very much filling that container, and they're ignoring the fact that they're being touched on all sides by other cells. Things that can cause these hits to happen are excessive heat, radiation, chemicals, cigarette smoke, sometimes just your genetics. You just get cruddy genetics sometimes. Pollution can actually cause these things as well. Lots of different things can cause cancer to happen. Lots and lots of different things. When we're talking about cancer specifically, we have 2 main types, which is benign or malignant. Whenever we talk about something being benign, that's going to not really matter. Like, yeah, it's abnormal, but it's not going to cause serious problems. A lot of times, like dogs and cats will get fatty tumors that are just a little mass of fat, just creates a little ball under the skin and no big deal. A malignant tumor, this is the bad stuff. This is when cells are going to lose attachment to nearby cells and then they travel throughout your body and then they start more tumors all over, and that's called metastasis. So if you ever hear about an individual having cancer and going metastatic, that means that that cancer has left that part of the body and it has spread to many, many, many different parts of the body because those cells don't really care anymore. They've broken off. They're not following the cell rules. They're carried by blood and lymph all over the body, and they can infect other tissues. So when we are doing treatments for these different cancers, the 2 main things that we do are radiation and chemotherapy. What radiation does is it kills rapidly dividing cells. So as you can see in the middle there is that beautiful woman that is going bald and she has a wig. And that's because your hair follicles are also rapidly dividing cells, as is your skin and like the lining of your GI system. So radiation will cause vomiting. Sometimes it can cause open sores on your skin, and it can also cause you to go bald as well because it's killing all cells in your body that are rapidly dividing. Chemotherapy is going to stop DNA replication in all of your cells. So this also can have some different side effects. It can make you very, very nauseous and those different things because your body is just not dividing cells. Even if you have normal cells that are damaged, it's not going to be able to divide them for that period of time. And that's one of the reasons why we say that people undergoing these treatments are immunocompromised, because your immune system can't make new cells to keep up with threats. Even like a cold or something like that can be very, very dangerous for these people. When we're looking at new drugs and new ways to fight cancers, we're looking at targeting enzymes and different genes that we only find in cancer cells. So we have some different treatments. Some of them work really, really well in some people, and some of them don't. And we're still trying to figure that out, to be honest. And it's a really, really long, hard process, but doing the best that we can. It is kind of like the big scary disease that we still have, and it's just caused because of some of these damages that are happening within our cells. All right, so that is the notes for this. So I'm going to turn off the recording.